Our fourth session then, and our first session of today, we've got some really great talks lined up. I am going to hand it off to Carly Nilsson with the State Water Board to moderate this session. Before I do that though, Carly, I wanted to give you a proper introduction. So Carly has her BS in biology and has been a researcher, consultant, and fellow for water quality protection. She has worked for the water boards for almost 15 years, most recently taking on a new position at the State Water Board in our office in AWEMA as the co-lead for the freshwater and estuarine harmful algal bloom program. And with that, Carly, I'm going to hand off to you.
Okay. Hi, everyone. I'm really excited to be here. We're doing session 4 today, and so we have 4 awesome speakers, so settle in, get your coffee, get comfortable. I think today it's going to be kind of a unique, different kind of approach. We're going to talk a little bit about endangered and threatened species, some fish, and we're going to talk about some algae, which is right up my alley. So with that, I'm going to introduce our first speaker. Gary Chin is a second-year PhD student in evolution, ecology, and organismal biology at UC Riverside, advised by Dr. Helen Reagan and Dr. Kurt Anderson. His research interests are in freshwater ecology, ecosystem health, and ecosystem services. His current dissertation work is on evaluating the ecological sustainability and governance structure of the Santa Ana River watershed in Southern California. Specifically, he's focused on studying the current and future ability of the watershed to deliver ecosystem services, sustainability, and equitably. In his free time, he enjoys cooking, exercising, and attending music festivals. So I'm going to hand it over to you, Gary. Don't forget, guys, that you can send in your questions during the presentation in the Q&A. With that, send it to you.
Well, hello, everyone. My name is Gary Chin, and I'm a second-year PhD student, as Angela said, in evolution, ecology, and organismal biology at UC Riverside. And today I will be talking about an assessment of threats to endangered and threatened freshwater species in California that I completed with members of my lab. And for the rest of this presentation, I'll be shortening endangered and threatened to ENT. So to begin, freshwater ecosystems supports high biodiversity through its variety of floodplain flow regimes. When we look at freshwater ecosystems, it's not just the free-flowing stream, but also the adjacent areas that are flooded at varying timescales. So looking at this figure from Poff et al., we can imagine that the riparian vegetation near the stream gets flooded regularly, perhaps annually, while the areas further away from the stream may only get flooded once every decade or century. And this wide range of flow regimes that exist in freshwater systems in turn supports a wide range of species and populations. Freshwater ecosystems contain as much as 12% of all known species, including a third of all vertebrate species, and approximately 40% of global fish biodiversity. And so a reason for this is the fact that this small amount of water in the world is not only responsible for sustaining wildlife, but also for sustaining human activities such as irrigation and municipal and industrial consumption. And this intense human demand for water and its associated stressors ranging from agricultural runoff and flow regime modification from dams to oil spills affecting wetlands puts a huge strain on such a limited and dwindling resource. Yet, as we all know, considerably less attention and research is put on freshwater systems despite water insecurity currently impacting more than 2.5 billion people. And by the year 2030, human water demand is expected to exceed reliable supply by 40%. And just to reiterate the fact that freshwater ecosystems are biodiversity hotspots that are disproportionately affected. So when we look at this graphic from Reed et al. 2019, it shows the trend over time of the Worldwide Fund for Nature's Living Planet Index, which measures vertebrate population trend data for the freshwater, terrestrial, and marine realms. And here we can see that there is a remarkable index decrease in freshwater species relative to terrestrial and marine species within the last 20 or so years. And here you can see this graphic. So Dudgeon et al. Provides us with a useful framework in thinking about freshwater biodiversity threat. So what this figure shows is that we can categorize the most imminent threats to freshwater biodiversity into 5 categories, which are overexploitation, water pollution, habitat degradation, species invasion, and flow modification. And then you'll see all these arrows that connect those 5 threats. And that suggests that these 5 threats are strongly linked with each other. And therefore, conservation efforts have to balance and attempt to address all of these issues simultaneously. So only finding solutions to 1 or 2 of these threats at a time is inadequate in addressing the larger freshwater biodiversity crisis. And just by looking at the very nature of these threats, it really highlights the fact that freshwater is a resource that humans constantly extract, divert, contain, and contaminate in ways that compromises its ability to support its immense biodiversity. And in all the places where freshwater systems are threatened, it is especially precarious in Mediterranean climates. So outside the Mediterranean Sea region, Mediterranean climates can be found in Southwest and South Australia, the Western Cape of South Africa, Central Chile, and California and Northern Baja California. And with Mediterranean climates, they are characterized by highly seasonal and interannual variation in runoff and flow with long periods of aridity and isolation. Given these conditions, Mediterranean climates suffer from extensive land use change related to agriculture and urbanization, as well as extensive hydrological modification in the form of dams and reservoirs to protect human settlements against the flooding and drought characteristic to Mediterranean climates. For my research, we decided to focus on the state of freshwater biodiversity here in California. And with California is one of the few places where 5 climate types occur in close proximity. The desert, cool interior, highland, and steppe climates border a small region of Mediterranean climate and all contribute to the diverse biome in the state. The mild and winter rain of the Mediterranean climate in particular supports some of the highest species richest in the state. And these conditions also coincide with the highest human population density out of the 5 Mediterranean regions, along with high rates of endemism and trends associated with high freshwater biodiversity threat, such as high human water demand, massive urban growth, and extensive agricultural industry. And therefore, when we look at California, it really provides a useful case study for other places that are currently or will undergo intensifying freshwater biodiversity threat. And within California, there are 157 federally and state listed endangered and threatened, or ENT, animal species. Some examples of these species include the giant kangaroo rat, the San Bruno elfin butterfly, and the tidewater goby. Regarding these 157 species, we focused on 3 key research questions. So number 1, first, how many of the 157 species are freshwater species, meaning they rely on freshwater hydrological processes to live? Second, which are the most threatened taxonomic groups within those 157 species? And finally, third, what is the relative prominence of different threats across different habitat types? To answer those questions, we searched through online resources like the IUCN Red List website, NatureServe, and the California Department of Fish and Wildlife website to fill out an evaluation sheet for each of those species. So here is an example evaluation sheet for the San Bernardino kangaroo rat. At the very top, you'll see the at-risk classification. So FE for federally endangered, PCE for proposed state endangered, and so on. Below that, you'll see its risk factors, which we identified and evaluated using the IUCN Red List Threat Classification Scheme. So on the very first column, you'll see the specific threat category. And then next to that is the degree of threat and spatial scale, both rated as high, moderate, and low. After that is the temporal scale of the threat, whether it is short or long term. And finally, a specific description of the threat. So for example, if you look at the very first row of the risk factors, you'll see that we found that the kangaroo rat is affected by natural system modification, specifically flood control structures and operations and groundwater pumping. And we rated it as a long-term high-level threat throughout much of its range. And under those risk factors, you'll see that there's information about the species habitat association, spatial scale, functional group, generation time, and lifespan, if any of those are available. And once we completed evaluation sheets for each of those species, we aggregated and analyzed the information and had these key findings. Regarding the first question of how many of the 157 species are freshwater species, which again rely on freshwater hydrological processes to live, we found that most ENT species in the state are freshwater species at 64.4% compared to terrestrial at 28% and marine at 7.6%. And then on to the second question. So what are the most threatened taxonomic groups among the 157 species? So here's a bar chart with the taxonomic groups on the y-axis and the number of species on the x-axis. You'll see that the most threatened taxonomic group is fish, followed by mammals and birds. And given that we all know that freshwater species are the most threatened relative to terrestrial and marine species, and that all the fish species that we assess are freshwater, then this isn't really too surprising that fish are the most threatened. However, it's also important to be mindful of the fact that there may be some taxonomic bias in which species are actually listed on federal and state ENT species lists. We can also break down the freshwater, terrestrial, and marine habitat categories in this figure into specific floodplains. So in this iteration of the figure, the solid colors correspond to freshwater habitats, while the textured colors correspond to non-freshwater habitats. And some things that I noticed from this figure are that a sizable portion of the bird species are riparian, while a majority of the amphibians are aquatic, while a majority of the other invertebrate category, which leaves out insects, live in vernal pools. And then finally, moving on to the last question. So what is the relative prominence of different threats across different habitat types? And here is another bar chart with every assessed threat category on the y-axis with the number of species on the x-axis. So freshwater species represented here in light blue make up a majority of the species affected in nearly all the threats compared to terrestrial and marine species. And what's interesting here is that while altered hydrology through dams, reservoirs, and levees is the only threat directly linked to freshwater species are affected by every single assessed threat. The top three threats across all habitat types are habitat loss, invasive species, and climate change. However, when we look at just freshwater species, the top three threats are habitat loss, altered hydrology, and invasive species. And these threats, along with pollution and overexploitation, form the basis of the framework on freshwater biodiversity threat by Degenet et al. That I showed you earlier in this presentation. And again, we can break down the freshwater, terrestrial, and marine habitats into specific floodplains in this figure as well. In this iteration of the figure, we can see that while species living in freshwater aquatic habitats, represented in light blue here again, make up a sizable portion of the species affected in most of the threats, the species represented by the other freshwater associated habitats like riparian vegetation in yellow and vernal pools in red make up a sizable portion of the species affected as well. So just to recap, the key findings from our research are that first, most ENT species in California are freshwater species. Second, the most threatened taxonomic groups are fish, followed by mammals and birds. And finally, threats are most prominent in freshwater species when we compare it to terrestrial and marine species. And seeing the extent of threats to freshwater species in California highlights the need to find solutions to address multiple threats simultaneously, as Dudgeon et al. Suggests, and manage the trade-offs between ecological and societal needs, ensuring that there continues to be an equitable and sustainable delivery of freshwater ecosystem services. Future work should consider the spatial extent of threats to endangered and threatened freshwater species within California. Study whether geographic location and regional water allocation are significant factors in assessing threat to freshwater species. Third, conduct a similar analysis of threats to ENT plant species in California. And finally, conduct similar analyses of ENT freshwater species in other states so that we can develop a broader sense of the spatial, ecological, and societal extent of freshwater biodiversity threat. And with that, I'd like to acknowledge my PhD advisors, Dr. Helen Regan and Dr. Kurt Anderson, and our lab's former master's student, Anna, and former undergraduate researchers Leo and Eamon, who collaborated with me on this research project. In addition, I'd like to thank my department faculty and fellow graduate students for their constant help and support. And finally, I'd like to thank you all for listening. I'll take questions now.
Okay, thank you so much for that really great presentation, Gary. For all of you guys, please submit your questions for the Q&A. We do already have a question. How does aerial spraying rate as a threat for West Nile carrying culex using BTI or the OP Temphophos, for example?
What was that species again? So I actually have the spreadsheet of all the species that I looked at, so I can actually see specifically for that species if we looked at it or what the threat is.
Temphophos.
Could you type it in the chat just so I can?
Yeah.
Thank you.
And let's see.
It doesn't appear that we looked at that particular species in our analysis.
Okay. We have another question. We'll comment first. Great presentation. Just curious if you know what types of water pollution pose the greatest threat to ENT species?
For ENT species, from what we could tell, a significant source of the pollution within the state are pesticides and rodenticides. So particularly within more rural areas that are not necessarily under the purview of state and federal regulations when it comes to pollution within the state. So for example, like one threat that is emerging within the state is illicit agriculture. So specifically for cannabis and marijuana, that affects some more inland streams where there's unregulated use of pesticides and rodenticides that are getting into the water and affecting species that would normally be unaffected by pollution.
Okay. I had a clarification that it's culex as in the mosquitoes for the species. So I don't know if you want to check that really quick for me, Gary.
Oh yeah, sure. So just to clarify, the species that we looked at within this analysis are either listed as threatened and endangered in the federal list and the California list. So when I'm looking at the insects that we assess, they consist primarily of beetles and butterflies, but no mosquitoes.
Okay, excellent. I don't see any more questions coming in. I had a quick question for you. Do you think this kind of research, similar research, is going on anywhere else in the world in terms of these Mediterranean climates? And if they're kind of coming up with some of the same answers that you are in terms of what is most affected?
We're not aware of any similar analyses of endangered and threatened freshwater species in the other Mediterranean climates, but we mostly chose this to focus on California, given that we do live here. So there is some relevance to our own daily lives, but also the fact that there is just so much information available that might not be available in the other Mediterranean climates. So again, we had access to the IUCN Red List, NatureServe, as well as the California Department of Fish and Wildlife website, which in itself was so helpful in getting the information that we needed. So, but it would be definitely interesting to look at the other Mediterranean climates as well in the future.
Okay, I think we have time for one more question. What information or paper did you use to cite pesticides and insecticides as the greatest harm to T&E species?
So this isn't coming from any specific paper, but it's just coming from our own experience reading through a lot of the reports that were coming in. So just based on our observations, pollution through pesticides and rodenticides seem to be the most common cause of pollution threat. So in particular, a lot of the reports that we were using are the 5-year plans from the U.S. Department of Fish and Wildlife, and they'll list all the threats and then give specific narrative details about what that particular threat is. So, but it would be interesting, again, to actually look at what are the sources of pollution, just to give a specific number of how many species are affected by rodenticides versus pesticides versus some other chemical in the water.
Okay, thank you. That was an amazing presentation. I think we're at time right now. There's one more question that I think we'll send you to answer independent of this. And I think everyone can reach you by contacting the swamp email or Shuka directly. But that was awesome. Thank you so much for your time, Gary. I really appreciate it.
Okay. All right. Well, thank you so much.
And with that, I think we'll move on to our next presenter. And that was actually a great segue, especially talking about fish is one of the most threatened taxonomic groups. So now I'm going to introduce Steve Blumenshine, and he's a biology professor and director of research and development for the California Water Institute, both at CSU Fresno. His research program focuses on inland surface waters as habitats, primarily for fish. His current focus is the San Joaquin River Restoration Program, where he sustained work on juvenile Chinook salmon ecology in the system through a combination of empirical approaches, fish bioenergetics models, and coupled ecosystem simulation models. Earlier this year, he was fortunate to start a new research front on the C. Wandel Lake Change Collaborative Project in Lake Constance, which borders Germany, Switzerland, and Austria. And with that, I'll pass it over to Steve.
Hi, Carly. Thanks. Yeah, that was a great and important topic, Gary. And Carly's right. It set us up well here for what I want to cover. So I'm sure that many runs of Chinook salmon in California are on your list. And so what I want to cover, and I want to clarify one of the terms in my title, is macroinvertebrate diversity. So given that Gary covered some taxonomic aspects of these threatened species in California and the Mediterranean climate, our focus on macroinvertebrate diversity is really in the distribution of their abundance over space and time in juvenile Chinook salmon habitat in the San Joaquin River. So what we're focusing on, and again, a good carryover from Gary's talk, is with the San Joaquin River Restoration Program, which is a multi-year, multi-agency effort to restore this southernmost population of spring run Chinook salmon. So in this topic that I want to cover today about the San Joaquin River Restoration Project, for the material that follows, why does it matter? Well, there's goals for this restoration to meet the main objective of a sustainable population of Chinook salmon that doesn't need wholesale inputs from hatchery-raised juveniles and adult broodstock, for example. And in order to meet some of those restoration goals, we need to understand what's the carrying capacity of the system and how does that vary in that juvenile rearing area over time and space. Okay, so in California, many of the historical salmon runs were quite extensive and went into foothill and mountain areas. But from this map that many of us have seen, that many of the reproductive areas and juvenile rearing habitats for Chinook and other salmonids in California are greatly truncated, such that dams preclude their return to natural spawning areas. So in the last century, that was a rather abrupt and serious habitat change and interruption to their life history needs. So what I'm showing in the bottom left picture here is an aerial photograph of Friant Dam, which creates Millerton Reservoir here. And so what's allocated after what goes down canals to feed agricultural interest is this little trickle of a river, that's the San Joaquin. And here and about 20 kilometers downriver is the current rearing area for Chinook salmon in our study system. So closer to the ground, what we could do is take a lesson from Peter Moyle and take a look at the current landscape that we have, for example, and it mentions Mediterranean climate regions. So it's the new reality for streams in California and other Mediterranean climate regions. So Gary, there's more of a connection than I initially realized. And the fact is with restoration programs, we can't go back to what was before human alteration of hydrology, for example, in these important habitats. So we have to work with what we have. And so our focus and our approach in our role in the restoration effort is to recognize this altered hydrology as well as prospective changes due to climate change, which are impending. And so overall, we look at the thermal experience for the fish. And that is really a thermodynamic approach, looking at energy gain through prey consumption and energy costs, say through swimming against the velocity of the river, and the metabolic energy, which are based on the temperatures of the water. So we look at this in a big picture is the scope for production. What's the potential production for juvenile salmon cohorts given the thermal and other energy regimes in the rearing area? So let's take a look at a table from the Fisheries Management Work Plan for the San Joaquin River Restoration. And we kind of zoom on this table, which is based on HSI or Habitat Suitability Index models. But that doesn't really jive with our approach we've used for years and years for a lot of different fish populations, in which those are based on thermodynamic principles and trophic bases for production. So for example, one of the objectives for juvenile growth rates of spring run is 0.4 grams per day for spring run or fall run, or 0.07 grams per day for those that are still there in the summer. And we've argued with them or pointed out that this metric is not really energetically based, and there's some issues with it. For example, if we consider this metric, 0.4 grams per day, well, that really matters. These small fish grow very rapidly, we find. And if we're looking at a 2-gram fish, 0.4 grams per day is 20% growth per day. That's fantastic, almost impossible. But for a 10-gram fish, that's just 4% per day. That's not bad. But if we get up, say, to a 20 or 40-gram fish, 0.4 grams per day isn't that great. And so we argue that it's not really connecting with the carrying capacity of the habitat and the scope for production. And we're asking for some consideration of some changes of the goals of the restoration program to better look at that approach. So for example, using this 0.4 grams per day growth rate as a baseline, we find from a lot of data that very few fish meet or exceed that 0.4 growth rate in grams per day, but we find the growth rates on other measures are quite good actually. Rather, we prefer to look at the bigger picture and have an alarmic approach to growth rates such that small fish, like puppies and human babies, can grow very fast, but as they grow, their growth rates, again, based on temperature, tends to diminish. So we have an interaction between temperature and the size of the organism in assessing this scope for growth. And we use this value omega, which standardizes the growth rate across these different sizes, recognizing that very small fish have a very high capacity for fast growth, and that diminishes as the fish grow in size. So this omega value, for example, standardizes growth rate. So we have a 1 gram fish growing at 1% specific growth rate, mass per day, and everything else, bigger fish, for example, are relative to that, as well as their specific growth rates. So that standardizes that allometric interaction we have between temperature and size, for example. Okay, so our focus is on looking at the thermal environment plus prey energy. Those are both based on thermodynamic principles. So for example, we know that the thermal environment in the rearing habitat is quite variable. Areas that are blue are cool over time and space, the two X and Y axes here. But as we go further downriver over time, things get a little hot. So in our simulations of growth and the habitat, we consider this very dynamic thermal landscape. And then we have the prey energy. And if we have a lot of prey energy, fish will do okay because they could fuel that furnace that's burning pretty hot in these warmer temperature times and places, for example. So overall, we coupled in a prior iteration of our research, which is ongoing over a number of years, empirical information, collecting data like this thermal history and the macroinvertebrates with simulations using tools like bioenergetics, which is an IBM or individual-based model, and InStream, which is a habitat bioenergetics coupled model. So our overall approach, I'm not going to walk you through all this, but in our proposals, we're looking at water allocation. So we know that surface water is scarce and there's a lot of competing users for water in California. So we want to look at the efficiency of water allocation for the restoration based on bioenergetic principles to better assess juvenile Chinook habitat and their needs to meet these restoration goals. So the currency we look at are energy units like calories per grams of fish, and we use these coupled type models to predict some different scenarios. So the InStream model, for example, there's a new version 7 that we worked with Steve Railsback on that, and that has a lot of physical variables as inputs. And then finally, we get down to prey quantity here, a biological input that fuels the metabolic furnace that the fish need for growth. And with the simulations, we could estimate growth, survival, and condition of juveniles in any cohort. So I'm showing you this. It's a lot of data, but the main point here is that this thermal environment is quite variable. For example, we can't just say that we have a drought year because we know from the last big drought time that the drought cycles aren't running in high precision. They could be early or late, and they differ quite a bit from normal hydrologic cycles in the river, for example. And we keep track in our simulations of what thermal environment individual and cohorts of fish are experiencing by tracking how temperature in the river changes over the cohort days. So when they're born, approximately November 1, at different locations in the rearing area. So we kind of interpolate their thermal experience as they undergo successive days in the river and even move downriver, for example. So what we could do in our simulations is to incorporate drought years or normal hydrology. So based on the river restoration program, there's designations of water years going from normal wet to critical low. We could couple that with different baseline abundances of prey. I'm going to skip this and move on. I think it's just showing some of our empirical data showing that temperature isn't that related to fish growth. So some other factors must affect growth rate. So with that, we could take these scenarios of water habitat and prey abundance I have on the right part of the schematic, but the inputs are the prey abundances and availability, and that's the macroinvertebrate diversity part I had in the title. So we need to consider a diversity of habitat conditions and where the fish might forage. Search mode is when they look along the bottom of the river for benthic prey. Drift is most traditionally studied to assess habitat. But then there's macrophyte habitat. And there's a lot of that in this rearing area of the San Joaquin River. And that's not very common to salmon habitat, so it's really understudied. And then we have several methods to estimate growth rates. And we can couple all this information to calibrate some best fit models and undergo more scenarios of combinations of the type of water year, and the amount of prey. So for example, the macroinvertebrate diversity, if we look at drift samples, which is what most people look at when they assess juvenile salmon habitat, we get lots of tiny prey like this. These are little crustaceans like copepods and cladocerans. And so that's like breadcrumbs. If you're a forager, it's not very efficient. But when we look in this understudied macrophyte habitat, oh my gosh, that's where all the big stuff is hiding. And this is a different packaging of energy that, if we follow optimal foraging theory, is more energetically beneficial, we'd suggest, than foraging in this crumb-like environment here versus having a whole piece of the rice cake or bread in this type of habitat. So we're trying to figure out how to incorporate these variables and these differences in the prey among habitats in our scope for production estimates. And then just a few results that we have so far. Overall, it's showing that there is some variation in drift abundance, say in time and space. And then in the macrophyte habitat, I'm going to have to skip to this one, we see different invertebrate abundances among different plant types. So that makes our analyses a little difficult. We have to account for degrees of freedom there. And then overall, we see a significant month and site interaction for macroinvertebrate abundance in different months and different sites along the river. So that's going to mean that we need to chop up our simulations in more tightly spatial regimes in the river. Okay, so to assess how the fish relate to those different habitat types, we use stable isotopes and mixing models. We have data from the invertebrates. We just got data yesterday from the fish, so I can't include them here. But one thing we see in the clouds of the stable isotope signatures from carbon and nitrogen for invertebrates is that we see a nice downriver signal, which is really good because then we can kind of assess where the fish got their nitrogen and carbon along the river reach. Okay, so I'm told I have 1 minute left. So we use mixing models to look at the probability of the fish and their carbon and nitrogen and what type of prey it came from, for example. So overall, we're really focused on this fish habitat relationships from a bioenergetics point of view and the heterogeneity of the habitat and the prey energy that they contain. So we're focused on this thermal and energy source considerations and using integrative simulation-based processes and interdisciplinary groups with lots of different expertises. And so we want to take that theory and those simulation approaches coupled with the empirical data and help those to advise and guide some applied goals like Chinook salmon restoration. So finally, I want to thank a lot of people who've worked on this over the years, both agency and people in my lab group. So thanks, and I'm interested to see what questions you have.
Thank you so much, Steve. That was super interesting. We're still waiting for some questions to come in. So with that, I guess I can ask a question just kind of based on your research. Did you guys do any bioassessment on like the proper for macroinvertebrates at all to kind of couple with this, or was it just looking at the fish species?
Oh, we've spent lots and lots of effort on macroinvertebrate sampling and analysis in the lab. That's very labor intensive. Yeah. Okay.
And how would you envision kind of incorporating this kind of research into a restoration project that is specific to kind of fish habitat for some of these larger river systems?
Yeah, larger rivers are going to be a tough call. San Joaquin is big enough you can nearly walk all the way across it, but you need to consider, say, for the Sacramento River, the morphology of the habitat. So you're going to have a lot of volume per surface area, for example, with respect to the river margin and contact with any bottom habitat. And so you need to look at your prey energy sources appropriately and scale that to the morphology of the habitat, whether we have a low width, shallow river or a broader river that's very deep and high in volume.
Okay. And we're getting some other questions, so I'll read those to you. How can you test if fish are actively feeding in the macrophytes? Do you think macroinvertebrates can avoid fish predators in that habitat?
That's exactly what we're doing, and that's our new front now. So actively feeding in the macrophytes, we could do that chemically with the stable isotope signatures, but that's inferential. And just in the next couple of weeks, we're going to put out GoPro cameras to monitor fish movements, both the juvenile salmon and predators there. And then we're going to work with our agency colleagues to put in some minnow trap type systems to look at who's entering and exiting the macrophyte habitat.
Cool. And great talk. Do you have any data on macroinvertebrate production in addition to density diversity?
Hey, Kurt. Yeah, that's a great question. So, a lot of estimates of macroinvertebrate productions are like by size frequency method and things like that. So I don't have anyone in my lab dedicated to that exclusively, but maybe let's follow up with that. I'd like to hear from you and see where we could go with that, for example.
Awesome. I guess that's going to end it for the questions. Kurt says thank you. So I just want to thank you so much, Steve, for your time. It was great to hear a little bit about the fish species because I know we've been focusing so much on the bugs the last 2 days. So I really appreciate you taking the time to kind of share a different, lens with all of that.
Thanks, Carly.
Yeah. So with that, I'm going to go ahead and introduce our third speaker, Byron Furman. I might have butchered the last name. I apologize. Byron is an aquatic technology development scientist at CPRO Corporation, and he is the Northern California Director of the California Lake Management Society, commonly known as CALMS. He received his PhD in environmental systems from UC Merced, where he studied redox and oxygen control on water quality in California reservoirs. He also holds a BS in chemistry, an MBA, and an MS in environmental engineering sciences. He has previously worked as an instructor for an environmental engineering course on water quality and as the quality assurance manager at an environmental laboratory. And with that, I'm excited to introduce him because we're going to start talking about my jam, algae.
Thank you, Carly. Yeah, thanks for everyone for having me. I'm really excited to talk about nutrient and mineral cycling and just how it relates to aquatic ecosystem structure. For my job, I mostly work on aquatic technology, so just kind of trying to find new solutions to water quality problems or ways to go about managing water bodies. And so this talk is kind of derived from like the research that I do, but it's really just how can we use water quality and algae data to better inform management. And that can be whether it's preventative management, reactive management, or even just predictive management. So understanding there's going to be a problem before it occurs will give us a lot more tools to work with that. So I'll just start off with the basics of nutrient management. So most water bodies are naturally oligotrophic. So they're pretty blue, clear. You have a healthy food web. So you don't have excessive algae because the algae growth is kind of controlled by the zooplankton and the fish and the birds and the wildlife. And so that's like your perfect, really functional food web. But with a lot of humans, we just love nutrients. It's just an integral part of our society, and especially of having large communities. So with things like intensive agriculture, stormwater, urban runoff, it's inevitably just leads to a lot of nutrient diversion into water bodies, and these nutrients will fuel huge algal blooms. So you'll have these naturally oligotrophic systems becoming mesotrophic, eutrophic, even hypereutrophic. And nitrogen and phosphorus are the main drivers of this algal growth. Some of the cyanobacteria can actually fix atmospheric nitrogen, so it makes it even tougher because you could try to limit the nitrogen, and then you might even just be stimulating the algae that produce toxins. The other thing is that algae need about 10 to 15 times as much nitrogen as phosphorus. This is pretty much all organic matter or all things that are alive. This really smart scientist, Redfield, he went around and studied a lot of different organic things and found that they all have a very conservative ratio of carbon to nitrogen to phosphorus that's pretty predictable. No matter whether you're a human, a bird, a cat, a dog, an insect, algae, whatever. And it's mostly because you just have so much protein and amino acids, which contain the nitrogen, whereas phosphorus is in DNA and ATP, and it's just less abundant in terms of overall mass. So overall, because phosphorus has been really limiting, and because cyanobacteria can get nitrogen from the atmosphere, just historically, nutrient mitigation to prevent harmful algal blooms has focused on reducing phosphorus. And I think it's important, and it's probably number one, but it's definitely not all that matters. There's a lot of other players in the game, and a lot of things that you can monitor and look at in order to determine, how your algal taxonomy is going to be just how, it's going to change or how it's going to be structured. And so the other players in the game that I would consider would be carbon, silicon, nitrogen, and iron. And these elements are important because algae have different chemical needs, and not all algae are created the same. So some of them are an integral part of the healthy food web. I would say green algae and diatoms are very important parts. And then some of them, they produce a lot of toxins. They produce taste and odor compounds. Cyanobacteria in particular, zooplankton don't like to eat them. And there's a lot of theories on the cyanotoxins, but one of the major theories is to avoid predation. So when, they're producing these toxins, zooplankton are less likely to eat them. So it's a competitive advantage to produce more and more toxins. And it's just a byproduct that humans have health consequences from it. So Carly, don't hate me, but I'm just going to generalize a little bit. And I'm going to say that green algae and diatoms are the good guys and the algae that we want to preferentially stimulate, not to obscene levels, but they're a very important part of our food web. And then the cyanobacteria are the bad guys. So not all of them are bad. Not all of them produce toxins, but we're learning every day about a brand new toxin we had no idea about before. So the more research that comes out, the more it leads freshwater water body managers to say, okay, a lot of cyanobacteria are causing a lot of problems. And so because they have these different chemical needs, we can just kind of keep it simple and say, good water chemistry is going to drive good algae. And if we can't control our water chemistry, we can monitor it at least, and we can know if our water chemistry is going to lead to good algae or if it's going to lead into bad algae and if we might have a problem. So starting off with some of the elements that I mentioned, carbon is really going to be CO2 and the form that it is in based on the pH. So because algae and cyanobacteria photosynthesize, they turn carbon dioxide into oxygen and get some energy from doing that. So based on the pH of the water is actually going to determine the form of carbon that they can uptake. So at a low pH, it's mostly in the form of carbonic acid, and that is in equilibrium with carbon dioxide. So if you look, carbon dioxide plus water is the same chemical formula as carbonic acid. So carbonic acid can dissociate into carbon dioxide, and that can poof out of the water. And so in general, at a low pH 2, you have a lower alkalinity. So you're going to have less overall carbon that's available for algae. And in these situations, green algae are good at carbon concentrating, a little bit better than cyanobacteria. And then diatoms have a lower carbon need overall. So they're going to be a little bit more competitively advantaged. And in general, you're going to have less productivity at lower pH because you'll have less carbon availability. Now at a medium or a high pH, you're going to have a lot more bicarbonate or even carbonate. And that's just because a factor of, it all being dissolved at a higher pH, you're going to have a higher alkalinity generally. And so you'll have a lot more carbonate minerals that are releasing this carbonate into the water column. So you're not as likely to be carbon limited, and therefore cyanobacteria tend to be a little more aggressive and a little bit more competitive under these circumstances. It's also a product of, they're not limited by anything. So they can take advantage of their other aggressive tendencies to get nitrogen or to get phosphorus. So silicon, I would say, is very underrated because diatoms are awesome, and they don't actually have to use carbon for their cell walls. So green algae make their cell walls out of cellulose. Cyanobacteria make them out of peptidoglycan, which is another sugar. But diatoms just polymerize silica. So if there is enough silica around for them, they actually have a huge competitive advantage in that aspect, that they don't need to photosynthesize and waste that energy to make cell walls. They can just do that with inorganic compounds like silica. And so if there is enough silica, they're stimulated. But we've decreased silica loading into a lot of water bodies by damming and just in general, by pumping a lot of nutrients in. you can have a big diatom bloom early on in the season, and they'll use up all the silica and crash, and then you won't have any silica for the rest of the season. Whereas before humans started pumping in all these nutrients, you had a much more progressive growth of diatoms, and they wouldn't deplete all of their silica resources immediately. And diatoms are really important because they make a lot of lipids, which are really energy dense. So they're like an excellent fuel source for driving a really healthy ecosystem. The zooplankton love them, the planktivorous fish love them, and they're great water quality indicators as well. So moving on to nitrogen, the classic way that people look at nitrogen for water quality is using an N to P ratio. And the simplistic thought is just, well, toxin producing cyanobacteria, a lot of them have heterocysts, and these are specialized cells that can fix nitrogen from the atmosphere. So if we have really low nitrogen and we have an abundance of phosphorus, all the good algae that can't fix nitrogen aren't going to be able to grow. But then all of our nitrogen fixing cyanobacteria, well, they can grow and they can take advantage of these conditions. And so you're going to select for them in these conditions. But that's kind of a simplification because there's two different forms of inorganic nitrogen that algae can use. So there's nitrate, which is the oxidized form, and diatoms require this form. Green algae like it under certain circumstances. They're pretty flexible. They can, use nitrate or ammonium. Whereas ammonium is the reduced form, and cyanobacteria really, really prefer this form. It's a lot easier to incorporate into proteins, and so it just gives them an energy advantage. Diatoms can't use the ammonium, and green algae can, they can use either form. So in general, the high nitrate to dissolved phosphorus is going to be a lot more helpful, and having a level like that is really going to suggest that, hey, I can grow the good diatoms or green algae preferentially to the cyanobacteria. Whereas if you have you have a high N to P ratio, but it's all ammonia form nitrogen, then that's not really helping you. It might just say, instead of having nitrogen fixing cyanobacteria, you're probably just going to have non-nitrogen fixing cyanobacteria. And so the last one I want to talk about is iron, which I would consider definitely underappreciated fuel for cyanobacteria growth. So nitrogen fixing cyanobacteria need an insane amount of iron. That molecule on the right-hand side is a cofactor in nitrogenase, which is the enzyme that fixes nitrogen. And it's like 60% iron by weight. So they need a ton of iron. And even non-nitrogen fixing cyanobacteria still have a higher iron requirement. And iron's kind of a tricky one because iron oxidizes very rapidly in the presence of oxygen. And so if you have oxygen in your water column, you're not going to have any dissolved iron around because it's going to precipitate out to the sediment. So in a shallow water body, now the cyanobacteria have these cool little tricks, and they can secrete these enzymes that will dissolve this iron in the oxidized form and turn it bioavailable. But they can't use those tricks in deep oxic lakes because they can regulate their buoyancy, but they can only travel like 20, maybe 30 feet max before they're kind of out of whack. So they can't go 50 feet down to access the sediment in a really deep lake. But a lot of these deep lakes, especially because of all the nutrient infusion, they're getting really limited, or they grow a lot of algae and they become anaerobic. And so when they do become anaerobic, they can release iron in the dissolved form. And because they're anaerobic, they don't have any dissolved oxygen in that bottom part of the reservoir. So then now the cyanobacteria can dive to that part of the water column, the hypolimnium, and access the iron from there. So the best way to not have the nitrogen fixing cyanobacteria is just to have an oxic deep lake and to avoid their access to the iron. But obviously you can't really control that. So it's more just about understanding what you do have. And the other thing about iron is that the iron oxides bind a lot of phosphorus. So when you do have no oxygen and iron reducing conditions, you poof off not only dissolved phosphorus, but you also poof off the dissolved iron as well. And so both of those are really good at stimulating those cyanobacteria blooms. And so here's like a little just diagram of what I would consider like the collusion between the normal bacteria in the sediment and the cyanobacteria up at the surface. So you have the normal bacteria at the sediment, and they're going to eat up organic matter. And if they eat up enough of that and use up all of the oxygen, then you'll have iron reducing conditions, and they're going to dissolve the iron by reducing it. And then the phosphorus that was bound to it gets poofed off as well. So now you have all the fuel to grow a cyanobacteria bloom. Now the cyanobacteria bloom is going to produce a lot of toxins and avoid predation by the zooplankton. So it's not going to get eaten, and instead it's just going to decay. And so, it lives out its life cycle, and it falls back to the sediment, and it returns all of that iron and phosphorus in the cells in the form of organic iron and phosphorus. And now that sediment bacteria can do the process all over again, and it can degrade that organic matter, release the phosphorus and the iron, and drive another cyanobacteria bloom. And so I'll just leave with this final little diagram, and this is just hypothetical, but it's kind of telling us how we can use, nitrogen and phosphorus abundance and ratios and forms, or we can use things like the abundance of silica and iron and the pH of our water body to understand how we had different algae communities throughout a season. So I'm a huge proponent of seasonal monitoring and starting that monitoring before you actually have an algae bloom to know what your baseline water chemistry is and what nutrients you have and what nutrients you don't have before the algae actually start to grow. And then if you track these algae throughout the season, which a lot of these agencies are doing, which, all of the data we're collecting is really great. And so it's a good way to kind of integrate everything together. And I would say, if you are having problems, but you're not looking at algae identification and kind of determining, what percent cyanobacteria, what percent green, what percent diatoms, even looking at, into the species level of cyanobacteria to see which ones are producing toxins or which ones have the ability to fix nitrogen. I would suggest doing this because it's so helpful to know how this algae community transitions throughout an entire season. And it's very classic to see a diatom community at the start of the season, and they'll use up all of the available silica if you have a lot of nutrients around. So they'll bloom and then they'll crash, and then you won't have very much silica for the rest of the season, especially in California where we get most of our rainfall during the winter time. So if you don't get much rainfall during the spring, summer, and fall, and then you have all of the runoff that comes in the wintertime, well, you regenerate your silica in the wintertime, but after your first bloom of diatoms, you probably won't see any throughout the rest of the season. And that just makes less competition for the cyanobacteria. So it gives them more preference. Some lakes, you could very easily correlate the amount of iron in like the depth that the cyanobacteria can access to your cyanobacteria abundance. And so you can probably, if you just look at it through one season, you can figure out, okay, when do we actually start releasing iron? And maybe it's only in one part of your lake or your reservoir, but then that cyanobacteria bloom that's triggered spreads to other parts of the lake as that community, migrates out to find a new niche for them. And so, just looking at simple things like the silica and the iron abundance throughout time can help a lot. If you track your nitrogen to phosphorus ratio, if you look at when you have nitrate versus when you have ammonia, this can really help. And so, all of these are just really ways that we can use the information we have at our fingertips to try and, better predict what's going to happen with our water bodies or to figure out some management strategies for them. So thank you everyone for your attention, and I'm happy to take any questions.
Thank you so much, Byron. I really appreciate you commenting about, monitoring before you actually have a bloom so you can actually implement some management strategies. Super important. We have a question. In a lake or river, do you monitor nutrients in subsurface or depth integrated samples?
That's a great question. If you have the ability, I love depth integrated. So, sampling every 3 to 5 feet, or if you're doing it in meters, classic is doing it every 3 meters. So you can get a surface sample every 3 meters down if it's pretty deep. And, when you do these kind of samplings, you'll tend to find that your surface, like if your water body is stratified and you have different layers, you'll tend to find that your surface, it's all pretty much the same. And then your hypolimnium is all the same. And then you have this really small transition zone, the metalimnium. And so if you have like a data sonde, then you can figure out where, you're in the surface water, the metalimnium and the hypolimnium, and then you can get away with taking 3 samples. But if you have active like nutrient or iron release, then you're going to have a higher concentration. The hypolimnium is kind of like stagnant. So if it's actively being released, you'll catch more of it down closer to the sediment as opposed to like the upper hypolimnium. So I guess that's a bad answer. It kind of like depends. But yeah, the more data you have, the better.
Awesome. Thanks. In a resource-constrained public agency managing a lake, what's the most effective yet affordable method for collecting the biological data, i.e. diatoms, green, and cyanos, needed for the seasonal pattern analysis? And what frequency would be best and most affordable?
I would say if you can justify it, having someone with a background in algae, like someone like you, like we have some experts here where I work that do cyanobacteria or just algae identification, and they're so quick. Like I'll look on the microscope, but I'm a chemist by training, so I'm not the one to know like, oh, this is this type of species of cyanobacteria, this type, or no, that's a green algae, or this is, pico cyanobacteria. I don't really know that stuff, but there's a lot of experts that do that would be able to, serve that capacity. And just with given a microscope and a centrifuge and, a water sample, they could tell you exactly what's in your water. I would say that's probably, like if it's a public agency that has multiple water bodies, it's a good investment to just have one person that does that for all of the water bodies. And then they can also do, research into like how these communities are changing. There's so many things you can do with that information versus sending it out to a lab. There are a lot of labs that do it, and our lab does it, but we're out in North Carolina, so there's I'm sure a lot that are closer to California that you could probably get a contract with to say, hey, we want to do 40 samples throughout the year at a discounted price. From my understanding, they're not extremely expensive because it's like 10 minutes for an expert to do that identification. I would say it would be one of those things where if your water body is dramatically changing, like community structure, like so frequently, you would want to start off doing it more frequently and then figure out when you can actually, like what period it shows like dramatic changes. I think you have a much better handle on this, but my perspective is like, an algae life cycle is like around a couple weeks. So if you monitor, monthly, you should see fairly decent changes in between that month because you won't have the same algae that was alive at the beginning and at the end of that month when you sample. If you can do every 2 weeks, I think that would be better. But I think, once a month throughout the period of, algae abundance, obviously you don't want to be, doing that before you have a ton of algae. That's just kind of more of a waste of money. But, start maybe April or so, April, May, June, July, August, September, October seems to be kind of reasonable.
Awesome. Yeah, and always feel free to reach out to our Swamp Hab program as well. We'd be happy to help with that. How do you suggest incorporating impacts from higher trophic level species such as sediment resuspension from carp?
That's a great question. It's not really in my area of expertise. I think I've primarily focused on deeper lakes. And I know that carp are a huge issue in like the shallow lakes. I work on Utah Lake a little bit, and they used to have a lot of aquatic weeds or just aquatic vegetation that reduce resuspension from all the waves and everything. And then they stocked it with a bunch of carp. Carp ate all of the plants, and then you have suspension from the carp and the waves or the wind. So it's definitely not ideal. I think that plays a lot into, like just nutrients getting into the water column. So it's a lot tougher than just having a stagnant water body with minimal resuspension versus, when carp are kicking things up, you can assume that you're not going to be phosphorus, nitrogen, or iron limited. But in that aspect, you could probably assume a little bit more silica. I think that's a great question. And that's why, holistic understanding of water bodies is very important. So, someone that focuses on just like the water chemistry and maybe a little bit of the algae here and there wouldn't have all the answers. Whereas, like someone that focuses on fisheries and someone that focuses on aquatic plants and, the whole big picture would be a lot better way to kind of understand these more complex ecosystems.
Awesome. Thanks. I think we have time for a couple more questions. Does deep lake cyanobacteria methylate mercury? Can your information be used to stop methylation?
So I don't believe the cyanobacteria are the ones methylating mercury. And that's just the iron reducing and sulfate reducing bacteria primarily. I did study that in grad school. It's not my focus right now from my recollection of what I was studying. I would say sulfate reducing bacteria were, in my opinion, they were the key methylators. So you have some iron reduce, and if you look at the redox ladder, so you have oxygen reducing conditions or oxic conditions, nitrate reducing, manganese reducing. So in those three, there aren't any known mercury methylators. Then you get down into iron reducing conditions, and there are some iron reducing bacteria that can methylate mercury, but they're not super prevalent. Then you have sulfate reducing conditions, and the sulfate reducing are the predominant genre of mercury methylators. And then you have methanogenic conditions, and they can methylate mercury, but they're also very good demethylators. And so I think one of the takeaways from what I remember was actually bloom dilution. So that concept is that under anaerobic conditions, you can have more mercury being methylated, but because you release more nutrients and you grow more algae, you actually have a higher algae to toxin ratio for when you're putting it into the food web. So if you just think of the amount of carbon I have in the water column versus the amount of methyl mercury I have in the water column, even if you have more methyl mercury, if you have 5 times as much carbon, then you're going to have more nutrients to toxins. So, there's kind of conflicting studies on things like oxygenation to repress mercury methylation, because maybe it would repress it by keeping it oxic. But then on the flip side, if you reduce your overall algal productivity, and then now the zooplankton have less to feed on and there's more methyl mercury in that algae, it can become a problem. So mercury was really a tough one. But yeah, if you want to send me an email, I'm happy to talk about that stuff. It was fun back in the day.
Awesome. In shallow lakes, what do you see as the best management strategy for controlling the effects of iron mineral reduction and phosphorus released from sediments?
Shallow lakes are tough. So I would say, you really just can't get away from it, because the cyanobacteria can always access the upper sediment and get that iron oxide. You can mitigate phosphorus chemically with different ways. I haven't seen oxygen being very effective. If anything, it's detrimental in shallow lakes because you're just stirring it up even more. So I'd hate to say that on a presentation like this where I wanted to talk about all the other things, but it comes down to phosphorus, chemical inactivation. I'm not the biggest fan of alum. And then I work for a company that sells phoslock, so I don't want to just sit here and be the chemical salesman. But yeah, I would look into like phosphorus mitigation. I guess iron's actually a decent. I've seen some studies where they added enough iron to, because you have oxic conditions and you're not going to have the iron dissolve, then you can actually kind of enhance the sediment binding capacity of phosphorus and keep it in that bound up state. But then another thing is that a lot of the shallow lakes I've worked on, they underestimate external loading. And so they'll think of, oh, okay, well, we get so much. And if you have carp and you have a lot of like resuspension, you do get a lot of phosphorus from the sediment. But if you don't, then you're probably getting a lot from your watershed and from like nutrient loading in like the early spring. It's like a huge factor. And so if you get all this phosphorus that gets dumped in before you're even monitoring it, you won't know because there's not enough light to drive that algal growth. And then, by the time the light hits, you have this, a ton of algal growth, or you have a lot of that phosphorus that's just like slightly bound up to the sediment, but stuck in the pore water. And so then the algae can access that. So I would say in a lot of shallow lakes, they kind of, you underestimate the loading from watersheds. But again, a complex question. So if you want,
yeah, we're getting heavy hitters today.
happy to talk about that more. But yeah, great questions.
I think there's time for one more, and then we'll have to pass. But have you ever seen evidence of bloom dilution before and/or ripple effect after an algae bloom?
And is this in relationship to mercury methylation?
I think the participant actually said that, oh yes, it is in relation to that.
So yeah, I have seen bloom dilution. My PhD advisor is Mark Butel, and so he looked at oxygenation in these twin lakes in Washington. So they had this just model system to study where they were these two geochemically very similar lakes, similar depth, and they oxygenated one of them and didn't oxygenate the other one. And they wanted to see, okay, with oxygen, you usually see lower methyl mercury. They saw that, but then they saw higher methyl mercury in the zooplankton and in the small fish. And mercury studies are tough because they take so long to actually translate up the food web. So to change the concentration in large fish, whether you're trying to reduce the concentration or whether you're trying to see if the concentration is increasing because of enhanced anaerobic conditions, it takes a long time. But they saw increased methyl mercury by dry weight ratios in the zooplankton and in the small fish in the oxygenated reservoir. And they linked that to the reduction in productivity. So when they oxygenated it, they stopped all of the nutrient release and they had lower algal abundance. So the zooplankton had less stuff to prey on. And then the organic matter that they're eating inevitably had higher methyl mercury to carbon ratios in it. So they kind of stocked up their food source with the toxins, unfortunately. And it's not, all lakes are different. So some oxygenation systems will stir up the sediment water interface, and then you'll release more nutrients. So in that aspect, maybe that would be the best. So you have oxic sediment water interface, no methyl mercury, but then you have more nutrients. But then that's a tough situation too, because, sometimes increased productivity leads to other increased toxins. But yeah, it's always fascinating to talk about.
Yeah, definitely. Well, thank you so much for your time, Byron, and for, helping answer and explain some of these difficult concepts. We really appreciate it. With that, we're going to move on to our last speaker in this session, Dr. Jamie Smith. She's a senior scientist at the Southern California Coastal Water Research Project, known as SCWRP, who studies the ecology of harmful algal blooms in aquatic systems. She wanted to highlight some of the project contributors. Dana Schultz is a senior research technician at SCWRP who specializes in HAB ecological studies. Rich Badness is an engineering geologist with the North Coast Regional Water Quality Control Board and has extensive experience with benthic cyanobacteria in Northern California. Dr. Emily Duncan is a senior environmental scientist with the Los Angeles Regional Water Quality Control Board and is the regional HAB response liaison. Dr. Susanna Thoreau is a senior scientist at SQUIRP who focuses on the development of molecular methods for bioassessment and the study of microbial communities. And with that, I'll pass it on to Jamie.
Great. Thank you, Carly. Appreciate the introduction. And again, I'd like to thank my co-authors for their contributions. I'm honored to be able to present on all of our behalves. So let's see. Oh, okay. That's right. They warned me there would be a lag. So today I'm talking about DNA-based methods for identifying toxin-producing cyanobacteria. I think Byron gave me a really great lead-in for some of the topics that I was going to cover today. But to kind of just take a step back, cyanobacterial HABs are a major issue in California. I'm sure many of you have heard about them, particularly in the last couple of years. There's been a lot of kind of high-profile news stories regarding harmful algal blooms. And California has really done a lot since 2016 to understand the extent and magnitude of the issue. And it truly is a statewide issue. I'm showing here on the left-hand side the 2020 HAB reports and advisories, and they span the entire state. And we have a number of different issues with different toxins. Microcystin has been a historical focus, but there are issues with other toxins like antitoxin and cylindrospermopsin, issues with multiple toxins simultaneously. And so this is a, let's see, this is a cross-cutting issue as far as beneficial uses are concerned. Harmful algal blooms are a cross-cutting issue that affect basically all of the core beneficial water uses, mainly due to the toxic secondary metabolites that they produce. Cyanobacteria have been around for 2 billion years, so they make all kinds of chemical compounds. They obviously cyanotoxins as well as taste and odor compounds and a lot of other things, like as Byron also mentioned in his talk, there's the more we look, the more we find different kind of toxic metabolites that cyanobacteria make. And so this obviously impacts, drinkable, fishable, swimmable, and aquatic life uses, as well as a lot of tribal uses, which I'm sure this will come up later in the tribal use panel today. But there's been a lot of study, but we still have a hard time really assessing the overall impacts on beneficial uses. And that's because there's a lot kind of going on when it comes to harmful algal blooms. So I just want to kind of put in your mind a framework for what makes a toxic algal bloom. So we have cyanotoxin production, right, which is the source of a lot of the issues, but that kind of requires both the presence of a toxigenic species as well as environmental conditions that promote toxin by those species. Toxigenic species don't just pump out toxin all the time. Toxin production is a biologically mediated process based on environmental cues. And so your nutrient conditions really and temperature and other factors really influence the toxigenic species to either ramp up or kind of ramp down their toxin production. And so understanding this issue is we kind of really need to look in these different boxes, right? So a large focus thus far has really been on cyanotoxin production and characterizing where we're seeing the toxins. But there's a lot less focus on identifying, the environmental conditions that lead to toxin production and the toxigenic species that we're actually dealing with. And really, to effectively manage cyanotoxin blooms, cyanotoxin producing blooms rather, we really need to kind of have this holistic approach where we're looking at all of these things because again, Byron gave me a really great lead-in. Depending on the species that you have and the environmental conditions, you can have very different kind of cyanotoxin outcomes. And today I'm going to really focus specifically on the presence of toxigenic species. So there's multiple ways that you can go about really characterizing your algal community. The most traditional way is microscopy. So you go out in your environment, you collect samples, you do morphology-based taxonomy, and then you can kind of get a sense of what's going on in your community. But this is actually a bit, challenging, and there is a high level of training that's needed to do this. I'm just showing here kind of on the left panel here, just to give you a sense of what these guys actually look like. So morphology-based taxonomy is possible with a trained expert, but it's challenging, and there's a number of issues. It actually can, depending on the level of taxonomy that you want to do, it actually can be quite time-consuming, as you might need a dedicated staff person to basically work through these samples, depending on how many you have coming in. And the other issue is you do get disagreement among taxonomic analysts, and so you can end up with variation depending on who's analyzing your samples. So another way of going about characterizing your community is a molecular-based method. So instead of collecting samples and doing morphological IDs with microscopy, you can basically collect the environmental DNA, extract that out, and run it through a biological bioinformatics pipeline on the computer, and you get taxonomic information that way. And kind of what I want to talk about today is that might be, a nice complement and a more streamlined and standardized approach, and potentially faster than kind of your traditional morphological IDs. So today I'm going to show two different case studies where we've used molecular barcoding to look at cyanobacteria in kind of cyanohab assessment scenarios. So talking about how you compare your DNA with your toxin analysis across diverse systems. This works across many different systems. Also identifying our problematic genre, and not so much environmental risk factors in this talk, but I think that's the ultimate goal of both of these studies that I'm going to talk about. And then evaluating, kind of to discussing how DNA can be a complementary tool to some of the more traditional or common HAB indicators like cyanotoxins. So to start, I'm going to talk about a case study, and this is a large contribution by Rich Fadness. So I just want to acknowledge all of his hard work in the field collecting all these samples. But in 2019, we have 18 samples collected throughout kind of the rivers up in Northern California, where we collected samples for cyanobacterial barcoding. And so we used a cyanobacteria-specific primer set and a curated cyanobacterial reference database that's based on Silva and GenBank, and as well as traditional microscopy and cyanotoxin measurements. And so what we found, or kind of what I want to show you, is that DNA-based methods do generally offer you a bit more of a refined view of your cyanobacterial taxonomy. So I have two arrows on this bar, and unfortunately the circles aren't showing up as they do in my version, but essentially what we're looking at here on the x-axis is what the microscopy ID for those samples were, and these are benthic mat samples too, to help you kind of, so these are kind of algal mats that sit on the benthos. They're usually filamentous typically, and so on the y-axis is the microscopy ID, and on the on the x-axis and on the y-axis is the DNA. The numbers are the number of samples with those IDs, and then we're looking at how they agree. So the purple arrow, what I wanted to bring your attention to is, yes, a lot of times we can get good agreement between our morphological ID via microscopy and our DNA based identification for genus level taxonomy. The red arrow pointing at the 33 is an interesting scenario. This is a scenario where we have Formidium and Microleus. These can look very similar. Their taxonomy was recently split, and the Microleus is more associated with toxic species, whereas the Formidium is not. And so in this case, these likely are more of an instance of this taxonomic change based on the toxin producing profiles of these different species. But with the DNA samples, once you have your DNA sequences, you can rerun them extremely easily through the pipeline, whereas with a microscopy sample, that's much harder to do. And so that's an advantage that DNA-based methods offer. They also offer, much deeper taxonomy. So here I'm showing just kind of a snapshot of what the diversity found in each of the river sites. And I wanted to also just call your attention that, these are, more like 10 plus genera, and each of these genera from a genomic standpoint, we've combined species. So there's usually generally multiple species within each genus. So we could even drill down deeper into this analysis if we wanted to and look at all of the different species of Anabaena or any of the other genera that we would be interested in. And so that was all of the cyanobacteria present in those mat samples. You can pull out just your toxigenic taxa, for example. So now I'm just showing toxigenic taxa. And this blue arrow here is indicating the area of the river where we found the most toxin detections. And so that, when we look at this, that corresponds with the highest presence of toxigenic taxa. So that can help kind of guide, you can use this tool in a way to kind of guide where you might have your biggest problems based on the biology. But it's not a perfect tool, and I don't want to sell it as your cure-all, but you can have instances. What I'm showing you here on the left is our toxin profiles from the mats, and on the right is the community composition. So you can have samples where, yes, you have toxin present and you have relatively similar communities, but then you can also have, as I highlight on the right, scenarios where you have dominance of one taxa associated with toxin, but then in another mat that's dominated that same genre, you don't have toxin. So it can help guide you, but it's not going to be a 100% there's toxin here, there's not toxin here. So paired sampling gives you the most complete picture of what's going on. So our second case study is on regional lakes in the LA area. So we did a regional lake survey back in 2020 where we looked at 17 different lakes and characterized the cyanobacterial community, the cyanotoxins present, and the basic environmental conditions. I'm going to give you a little snapshot of this study because there's not a ton of time left. But basically what we found was there were cyanotoxins in 7 of these lakes. And cyanotoxin genera that we found were more diverse than kind of your usual suspects. In lakes, a lot of times Microcystis kind of gets the bad rap as being the causative genre of microcystin, but in this case, that microcystis is present. So that's that light kind of green color. So if you look at Leg Lake, that lake had microcystin and was dominated by microcystis. But we have these other lakes where we have, relatively, detectable levels of microcystins, and you do have microcystis present, but there's other taxa, meaning, you can't just say, if I have this, I have that, right? Similarly, we have a couple of lakes with more novel toxins, things that haven't been studied as much. And so this is a nice example of, these are relatively understudied lakes, so we can get a sense of, well, what's the toxin present in these lakes and what are the potentially associated producers? And so if you have a monitoring program, you can start to kind of get an understanding of, if I start to see, in the instance of Machado, if I start to see Sphaeospermoptis, which is this purple, maybe I need to be concerned and I can guide my cyanotoxin sampling appropriately. And then I want to bring your attention to this last slide, my second to last slide, where these are lakes where we detected toxigenic genre and we had no toxin, right? So again, this is a good tool to understand these lakes have toxigenic production potential, but there was not toxin at this, point in time when we went to sample. But you might want to then pair this with observations of eutrophication or other indicators to say, are these high-risk lakes? Should I how should I adaptively kind of manage this system to make sure that I'm assessing my risk of cyanotoxins appropriately? So in conclusion, just some final thoughts. So DNA-based approaches, obviously I hope I made the case that they can offer a robust tool for morphological identification. You can get a high level of diversity and you can assess that effectively across your systems using DNA-based approaches. And I would highly recommend, pairing this and having like a holistic system by understanding your trouble species and the typical toxins that they produce, as well as the environmental conditions. You can really start to work on adaptively managing your system. And again, I think the speaker before me, Byron, mentioned, you want to start sampling before you have an issue so you can really understand how your system's changing. So again, understanding drivers requires consistent sampling, characterization of the toxin diversity, and the environmental conditions. And so with that, I really thank you for your attention and I look forward to any questions.
Thanks so much, Jamie. Really appreciate it. We do have a question. Since toxin production and or release can be at the species or strain taxonomic level, is there hope that DNA-based approaches can attain that resolution?
Oh, absolutely. I think that, I started kind of working on algae back in 2012, and it's just amazing the progression of molecular approaches just in that short period of time. And I think that, there's multiple ways you could use molecular approaches. There's tools where you could take, so what I'm showing you here is barcoding. So we're not saying this taxa is producing the toxin. It's just giving you kind of an idea of what's there. And you can develop kind of correlative relationships of like trying to understand who your problem taxa are. But there is starting to be approaches where you could sequence the community and pull out both the taxonomy and the toxin producing genes and link those two together to say, this is who's producing this strain or species is who's responsible or who's the common toxin producer in my lake. So I think that we're really, the possibilities of doing that are just increasing and are becoming more and more accessible over time.
Awesome. First of all, great presentation. Did you look at environmental conditions to see if there was any correlation with toxin production?
So we did do that. I didn't have time to put that into this presentation. And that's kind of I would say that's an active analysis that we're doing. From a high level, I think that we did see, correlations between more eutrophic systems, particularly in the lakes case study is where we did that. And I think you do see a general kind of correlation between your more eutrophic systems and the presence of toxin, but that's an ongoing analysis. So hopefully I'll be able to present more information in the future on that.
Excellent. And at what concentrations do these various toxins become harmful? We've done lots of sampling in streams across California this summer and have gotten a lot of hits, but I really want to figure out which of these sites would have harmful levels of these toxins.
Mm-hmm. And that's a great question. So from a human health perspective, in planktonic systems, the swamp program has great kind of guidance levels on what concentrations are harmful. There's the, your yellow, orange, red for the three main cyanotoxins of concern. And so I point you to the swamp FHABS program to look at that guidance from a human health perspective. From an aquatic life perspective, that's a more difficult kind of question to answer. And same thing for mats. I think that's an ongoing research. But generally, I think that what the common use is to use those human health thresholds right now to assess kind of your relative harmful versus not harmful or less harmful concentrations. So hopefully that helps. But that's an area of active research of really understanding your chronic versus your acute and all of those effects. So good question.
Awesome. And just to kind of piggyback on that, yeah, please look at the My Water Quality portal. That also has some information, which is on the State Water Board's website. So with that, I think that's all of our questions. And I just want to thank Jamie for her time today and all of the speakers in this, this session. It was super awesome. And with that, I will pass it on to Allie before our break.
Cool. Thank you so much, Carly. And thank you all. Those talks were all super interesting. It was nice to kind of get some biodiversity talks in there and then more focused in on cyano and algae and eDNA. So thank you all.